This video briefly discusses the data analysis and applications of flow cytometry. In data analysis, there are several plot choices: single color histogram, two color dot plot, two color contour plot, two color density plot. In single color histogram, fluorescence intensity versus the number of cells counted. Two color dot plot, Fi of parameter 1 versus Fi of parameter 2. Two color contour plot, concentric rings form around populations. The more dense the population, the closer the rings are to each other. In two color density plot, areas of higher density will have a different color than other areas. These are the diagrams of different plot types. Data analysis. Gating. Gating is in essence electronic window that sets upper and lower limits on the type and amount of material that passes through. Selection of only a certain population of cells for analysis on a plot. It allows the ability to look at parameters specific to only that subset. Interpretation of graph. An important tool for evaluating data is the dot plot. The instrument detects each cell as a point on an XY graph. This form of data presentation looks at two parameters of the sample at the same time. There are three common modes for dot plots: forward scatter versus side scatter, single color versus side scatter, two color fluorescence plot. Forward scatter versus side scatter to look at the distribution of cells based upon size and granularity. Single color versus side scatter to visualize the expression of the fluorescence of the cells. Two color fluorescence plot to differentiate between those cells that express only one of the particular fluorescent markers, those that express neither and those that express both. It is used to discriminate dead cells from the live ones that are expressing the desired fluorescence. How to differentiate dim and bright expression of an antigen. Dim cells are present more towards the origin. On x-axis, red color we can see. On y-axis, pink color we can see. In bright expression, cells are present away from the origin. On x-axis, green we can see, and y-axis, yellow we can see. What is unique in flow cytometry? It is multi-parametric. Rapid analysis of large number of cells are possible. Information at a single cell level. Detection of rare cell populations. Allows physical isolation of cells of interest. What is the uses or applications of flow cytometry? First of all, analysis, cells counting and cell sorting. Clinical application, HIV or AIDS, absolute CD4 count can be done. HLA-B27 assay in case of joint pain. Diagnosis and classification of hematological malignancies. Detection of MRD, S-phase fraction in solid tumors. In case of primary immune deficiency disorders, TB-NK analysis, that is complete lymphocyte subset panel. This panel may be useful for general immune status workup. In case of hemolytic anemia, reticulocyte count, PNH, and osmotic fragility assay can be done. Feto-maternal hemorrhage, treatment response in sickle cell anemia, fetal hemoglobin detection can be done. In bleeding disorders, platelet receptor assay, platelet function assay. Transfusion and transplant, CD34 stem cell count, residual WBC count in leukodepleted blood. Flowcytometry crossmatch, surface markers in PMN, monocyte cytokine responses.